All right, guys, let's go ahead and get into it. So I just did the Start Engine Live app. It's a relatively new thing that Start Engine created. I'd never done it before, and it was kind of cool. Not too many people showed up. I don't think people are looking at their Start Engine app during the course of the workday, but it gave me the opportunity to talk directly to people. And the truth of the matter is, I've done this presentation so many times. You can download it; it's everywhere. You can download on the website in the investor relations section. And what I realized was, just sort of having a dialogue and talking through some things is a lot more interesting. The truth of the matter is, I've had probably about 7 espressos, and I think I could run through a brick wall. And so what I want to do is let me talk through the story, and let's play a game. You guys write questions in the chat, and then I'm going to figure out how to incorporate it in the narrative in real time. And if I miss anything, we'll go back through it, and I'll address it at the end. So, everyone, thank you so much for attending. As always, it's really a pleasure. We've crowdfunded since 2019, and we've relied solely on you guys to fund what should not even exist. The truth of the matter is, Cytonics should have withered on the vine about 7 or 8 years ago. It is a miracle that a group of outsiders like ourselves, with no venture backing and no big pharma support, have been able to do what we're doing. And it's a testament to a grassroots effort. It's a testament to grit and just the human inability to give up on things that you believe in. And our founder, Dr. Gaetano Scuderi, is really the source of this this emotion, this ethos. And he founded the company back in 2006 while doing a sabbatical at Stanford, away from his surgical practice. And he decided at the time to try and tackle the most intractable, deleterious condition that all orthopedists see in their practices, and that's osteoarthritis. Osteo, not rheumatoid. And one of the reasons for that is that osteoarthritis is multifactorial. It's caused by a bunch of different things. It's multiphenotypic, meaning that it actually it can present in different ways. And the practicalities of running clinical trials to test disease-modifying therapies for osteoarthritis face many practical limitations. Now, that word disease modifying is very important because to date, the only treatments for osteoarthritis are palliative. They're Band-Aids, corticosteroids, NSAIDs. They just simply Band-Aid over pain and inflammation. They do not provide a durable, long-lasting effect that actually targets the root molecular pathogenesis of the disease. And what we have set out to do is exactly that. Now, as Nietzsche said, he who has a why can endure any how. And our why is simple. Big Pharma has failed to be able to develop a true disease-modifying asset over the last 30 years, and it's not for lack of trying. The clinical trial graveyard is littered with their failed attempts. But we know why, and we know what to do about it. And this is what gives us our edge. So what we have done is we have iterated on something beautiful, something natural, the ingenuity of nature. And it's this molecule, this protein called alpha-2 macroglobulin, or A2M. And A2M is a protein. It's produced in the liver and found in the blood, and it plays a natural role in blood clotting through a mechanism called protease inhibition. And proteases are enzymes that literally digest cartilage. They digest a lot of things. They play a natural role. I should clarify that they're not just the bad guys. They are responsible for protein turnover, and it's important. They play a natural role. It's important. Well, it turns out that the end of the molecular pathogenesis of osteoarthritis converges on this class of enzymes called proteases. And like I said, osteoarthritis is multifactorial. There are a host of proteases that destroy the cartilage. There are 2 major families called the MMPs, matrix metalloproteases, and ADMTases. And within each class, there are 2 proteases that are responsible for the majority of the cartilage damage. But there are also a host of more general proteases. So a truly effective disease-modifying therapy that converges on the protease digestion of the cartilage must act as a sniper rifle, but also a machine gun that can target these specific proteases and eliminate, in a broad spectrum fashion. And this is exactly what big pharma's failed to do. They failed to address all of these enzymes. Instead, when they've tried to develop protease inhibitors, they've tried the typical big pharma approaches. You have a single drug that is effective against a singular target for a singular effect. Why? Because it's tractable. It minimizes the potential off-target binding, which gives side effects, right? But the truth of the matter is, it's not going to cut it for osteoarthritis. It's a Medusa. You got to slay all the heads, right? So our approach converges on these multiple proteases. It is in direct opposition to big pharma's myopic approach to drug development. And we believe that nature has already created this molecule. It's the same story as insulin. Prior to 1982, when Eli Lilly and Genentech created Humalog, a recombinant, meaning a synthetic version of the insulin peptide, diabetics were treated with the insulin purified from the pancreases of, it was like dogs and pigs, but the naturally occurring form. After Lilly and Genentech were able to create the synthetic insulin, and they modified the gene sequence and they changed its properties. In the case of insulin, they changed its half-life. But they were able to iterate on something nature had already done and be able to create a optimized form that can be produced in the lab, scaled, dosed consistently, and distributed globally. And that's the holy grail, right, of a drug. You want to be able to achieve these things. So iterating and going from the natural form to a recombinant, meaning a synthetic, optimized form, is the same thing that Cytonics is doing. Back in 2006, Dr. Scuderi, while at Stanford, started this exploration. And it was mission-driven. What can he do about all of these patients suffering from osteoarthritis and their lives are absolutely debilitated? It's really hard when, your job day in and day out is to see people that they can't walk, and honestly, they'd rather just be dead. Like, that's extremely depressing. And if you're an empathetic person, it can take a really big psychological toll and affect his well-being. And Dr. Scary is a super type A person, really smart, didn't even go to college, did like 1 year college, took the MCATs. And just sort of, I guess you could say, a polymath, intellectual omnivore, interested in everything and extremely competent across multiple vectors. And so he started doing this research at Stanford and really surveyed the literature, the corpus of scientific data and medical peer-reviewed publications, and found this A2M and learned about it and learned that its mechanism is broad-spectrum protease inhibition, and identified it as a putative drug candidate. Maybe this is something that we could actually iterate on, right? So this is back in 2006. And before we ever went forward with the recombinant version, which talk about here in a second how we actually engineered it, we started with the natural version. And the first iteration of our technology was purifying out the natural A2M from the blood of patients, selectively enriching it, and injecting it directly into joints, called an intra-articular injection. And the first generation therapy, we call it the APIC system, APIC. APIC stands for autologous protease inhibitor concentrate. Autologous coming from self. Protease inhibitor, that's the A2M. Concentrate, self-explanatory. It's selectively enriched through a proprietary filtration process that actually just takes advantage of the fact that this A2M is big. It's 720 kilodaltons. Put in perspective, the insulin peptide, I think it was 191. I think it is 191 amino acids, tiny protein or peptide. So this is a monstrous protein. So literally through a sand and sieve mechanism on a molecular level, we're able to pull out this monster, right? And that's what led to our first technology being approved, I should say cleared, as a 510K medical device. A 510K medical device is something where the burden of proof from an efficacy standpoint is not very great. It really is a designation that you have developed a device that's substantially similar to a predicate device from a safety perspective. But we know from the clinical and commercial success of this APIC system, it's treated probably 10 to 20,000 patients in the United States, out-licensed to 2 distributors, one in the human market, one in the vet market. It works in horses, and ostensibly, or not ostensibly, purportedly other companion animals, something that we're still trying to expand into. the clinical and commercial success of this APIC therapy really is a testament to the power of A2M and its viability as a putative therapeutic modality to go forward and actually develop a biopharmaceutical. And that's what led us to Site 108. Encouraged by the APIC, encouraged by the natural A2M, we decided to employ the insulin strategy. And we employed a technique called proteomics, which is the study and engineering of proteins. So we used a high throughput shotgun proteomic approach to create hundreds of variants of the A2M. And what I mean by that is we went in, and let me actually go to this slide so you can visualize this thing. Okay, this is a pretty good visual. Yeah, all right, that's a good visual. We went in, and you can see you see the little red thing, and you see the blue globular structure, the blue, green, purple globular structure. That's the A2M. The little red things, and those two binding pockets, are the proteases. The A2M protein is tetrameric, meaning that there are 4 modular units. They're identical, and they come together to form this three-dimensional structure. And there are 2 binding pockets. So a 2-to-1 stoichiometric ratio between the A2M or the proteases and the A2M. So what we did is we made relatively few genetic modifications to the little binding pocket, the little V structure you see there on either top and bottom, 32 amino acids out of thousands, probably about 1,500 amino acids. Is about 4,500 base pairs or so. So something like that, probably 1,500 amino acids, something like that. So, it's a small portion, right? Those modifications are enough to make the protein, the A2M, stickier for the protease substrates, and significantly stickier. Basically, what we're doing is increasing its affinity, and the exceptional function compared to the natural A2M is measured in terms of protease inhibition. And if you can put a tiny amount of the synthetic form and have the same effect as a lot of the natural form, now you've developed something potent, right? And something that can be concentrated at supraphysiological levels, meaning much higher than naturally occurring, supraphysiological. And so what we did is we screened hundreds of these variants. Really, we created random mutations in this little binding pocket where we knew it wasn't completely random. We knew that certain motifs, certain amino acid sequences were responsible for the affinity of the A2M for its protease substrate. And so there was some intelligence behind it. But for all intents and purposes, screened hundreds of variants. And we settled on the 108th variant, which is why our lead drug candidate is called Cyte 108. CYT is an homage to the name of the company, and the 108th variant, which was the stickiest for the proteases, which has somewhere in between 200 to 300% more activity as an anti-protease agent compared to the naturally occurring form. So this was extremely encouraging, right? All these experiments were done in the lab, what's called in vitro, meaning literally in like petri dishes, right? We're trying to see which of these variants has the highest anti-protease activity, number one. Number two, it has to have that anti-protease activity against those 4 major proteases, but then also blast all the rest of them, right? So a broad spectrum, but also highly targeted and potent. It's really something very difficult to do. And one of the reasons, again, why big pharma's failed to be able to do it. So after we identified the 108th variant as the best thing to go forward with, we started our preclinical investigation. And the FDA's clinical trial process is divided into really 3 main parts. First is drug discovery and development in the preclinical stage. That's where you're going out, you're finding a putative therapeutic like the A2M, and you are doing some early experiments in the lab to establish a mechanism of action and to form a reasonable scientific basis to believe that, hey, this is worth pursuing in clinical trials. Then the second set of the pathway is the clinical trial pathway, broken into 3 phases: phase 1, phase 2, phase 3. They're successive. And the goal is to incrementally de-risk the development of the drug from both a safety and efficacy standpoint. In phase 1, you start in a relatively small patient population with the major burden of proving that it's safe. The FDA cares about safety, number one. Efficacy is irrelevant almost at this point. The primary endpoints are safety. We recently completed our phase 1 study with no adverse drug-related events, No drug-related adverse events reported. We're waiting for that statistical analysis to come out and the clinical study report to be finalized. That's going on like literally as we speak. Once you have established that safety in 20, 30 patients, our study was in 20 human patients. Now you go into phase 2, and now the burden is efficacy. You want to be able to establish a dose response, or at least see the efficacy of multiple doses, even if it's not linear, right? And you want to get a better idea of what the primary efficacy endpoints will look like when you scale into a phase 3. And that's a powered study, meaning that the number of trial participants will mathematically, theoretically, given the probability distributions that you expect. Remember from statistics, Gaussian curves, normal distributions. Let's say there's a line here, and on that line is, let's just call it effectiveness of the drug. It can be measured in multiple different ways. You have a placebo. It's going to be on the left-hand side, so that's, I think we're mirrored here, right? And it's going to have low effectiveness, and then on the right-hand side, high effectiveness. But remember, everything in nature is stochastic. It's not deterministic. You're going to have a probability distribution. Depending on the means of the efficacy, the average, and the variance, which defines the width of that Gaussian curve, that normal curve, you're going to have different probability distributions. Based on those distributions and the descriptive statistics that define them, you can back into the number of patients that theoretically you would need to be able to observe those distributions in the study. And in doing so, be able to theoretically gain statistical significance in the result. Statistical significance comes down to a false positive. What are the chances that we measure a positive signal in the placebo and or in either group, and it actually be due to chance? And basically, as a humanity, as the scientific community in humanity has decided that a p-value of 0.05 percent, or p equals 0.05 is what is acceptable to move forward and say, hey, this is a statistically significant result. So 5%, so 1 over 20. 1 over 20 events that are measured as classified as effective, classified as signal, classified as effective, are actually artifacts, are actually just due to variance, the stochastic nature of nature, right? So basically, the phase 3 is where you're trying to establish definitively that at this dose, you get statistically significant effects of your drug that are also clinically meaningful. This is one of the difficulties in osteoarthritis clinical trials, is that clinically meaningful in this context isn't a biomarker. It's not measuring the concentrations of cartilage, proteins. It's not looking at cartilage volume. It's not looking at any structural changes in the joint that could say, hey, it looks like this joint's getting healthier, right? It's what's reported by the patient, a PRO, patient-reported outcome. And in osteoarthritis, that's perception of pain, perception of inflammation. Now, can you imagine how much that varies even just through the day? When you wake up, you feel fundamentally different. Then halfway through the day, you've had coffee, you've been motion is lotion, you feel good at the end of the day. Now imagine over long periods of time collecting this data. The placebo effect is massive. The placebo burden's like 40 to 50%. Half of the signal you're measuring is actually just due to nothing, or perception, which is great from a patient treatment perspective, not great from a clinical trial perspective. This is one of the reasons why osteoarthritis clinical trials are so difficult. So you need a lot of patience, basically, to be able to back into what you think, what we think, would be the properly sized trial to be able to get a massive clinically meaningful score. So there's two things, right? We talked about statistical significance. We talked about clinically meaningful, right? Statistical significance measured due to chance. Clinically meaningful actually matters in a clinical setting. So basically what we're doing is we're incrementally de-risking every single step. So like I said, recently completed the dosing and all the patient follow-ups for the phase 1 clinical trial. No drug-related adverse events. Honestly, doesn't surprise me. Like I said, we mutated 0.1% of this molecule. The rest of it's exactly like you see in the human body. So not exactly surprising. especially given the fact that APIC works. So we're able to take A2M out of the blood and inject it exogenously into the joint where it's not found in those high concentrations. No problem. So not terribly surprised, but very, very important. Now, as we move forward, we're going to incrementally de-risk safety again, right? I mean, the FDA cares about safety every single time, and try and establish some meaningful signal of efficacy, even if it's not statistically significant. And this is going to be discussed with the FDA, but most likely what we're going to do next is a phase 1b/2a, right? And what that means is that, yes, you're going to go after safety again in a larger patient population, but not a properly powered patient population. Statistical significance is not what you're looking for. What you're looking for after you repeat safety is, can we measure an appreciable signal in a larger patient population and help define the actual endpoints we're going to use? Those patient-reported outcomes, there are multiple different ways to measure them. There's all kinds of scales of pain, all kinds of scales of inflammation and mobility or perceived mobility. The Western Ontario matrix, WOMAC score is one of them. There's COOS, there's VAS. They all stand for these different scoring systems that patients literally fill out. What's your pain like, right? Some of them are very discretized, 0, 1 through 10. Some of them are lines where you literally just in a continuous way say, I'm right here, right? Psychologically, it can be more effective to do it that way. So there are a lot of different things that you can measure. So now what we're going to do is incrementally de-risk the efficacy portion by exploring what is a good measurement, at what dose level will we get an appreciable, a meaningful response, and then take that information and scale it into a phase 2b, which can be properly powered. And then the information from the phase 2b inform the phase 3 clinical trial that is in thousands of patients. And ultimately, the parameters of that phase 3 clinical trial will inform the drug label. And the drug label is precisely what the physician will prescribe the patient: the dose frequency, the route of administration, and then all the other stuff about side effects, everything. Every single drug has 10,000 side effects, and it's simply because anything that the patient reports during the clinical trial, like a headache, random headache, it doesn't matter if it's related or not, anything they report, they could stub a toe and you could say, oh, pain. We had a patient get an itchy back. So it has nothing to do with anything. But that's why drugs have, a laundry list, right? So label, the phase 3, the parameters of the phase 3 will inform the label. So when thinking about drug development, you really got to work backwards. What commercially, how do I want to position this drug? Do I want it to be something that I give 10 times a week? Or do I need to design something with a longer half-life that I can give once every 6 months? What dose level? What's the route of administration? Is it an injection? If it's an injection, probably not going to be able to give it that frequently. How do physicians respond to this? Is this something that they will prescribe? How can you get insurance payers involved? Because one thing people don't really think about, who is the actual customer? The actual customer is not a patient. The actual customer is not a physician prescribing the drug. The actual customer is the PBM, the pharmacy benefit manager that represents the insurance company, because they're the ones who determine what drugs are on the formularies for the insurance companies and what drugs will get reimbursement. Do they have reimbursement codes? Will they be, a first-in-line therapy? So you have to strategically position the drug so that insurance payers will pick it up, physicians will want to give it. Ultimately, the patients just go along with that. How many times have you gone to a physician and said, actually, I've looked at the labels of these drugs, I would rather try this one. Everyone just goes with what is going to be prescribed by insurance and what the physician says works. So what we got to do is take that into account when we are trying to design the clinical trials, so that the phase 3 study will contain a label that is appropriate for how the drug will be commercially positioned. Will insurance payers want to pick it up? What we would want to be able to show is that the drug is so effective in the phase 3 clinical trial that it can actually obviate or delay the need for joint replacements. That's the holy grail. Because if you can do that, the trickle-down effect to the insurance payers' bottom lines is unbelievable, right? From a Medicare standpoint, from a private payer standpoint, from an employer payer standpoint, and osteoarthritis-related joint pain, back pain, it's the leading cause of missed work, right? If you can keep your employees healthy with an injection that's given maybe once every 6 months, once every 3 months, versus needing to pay for a massive surgery, that's huge, right? That's huge from an economics, a business perspective. It's huge from an insurance payer perspective, and it's huge from a patient well-being perspective. Last thing you want to do is have a joint replacement, which is end stage treatment. You're going to be shot up with all kinds of stuff before they send you to a joint replacement. Your insurance payer is not going to want to pay for your joint replacement until, everything under the sun, all the corticosteroids injections under the sun have been given to you, all the NSAIDs, you're pumped full of everything. And then finally you go for a joint replacement. It's not good stuff. A disease-modifying therapy for osteoarthritis is the holy grail of regenerative medicine. It's no easy feat. What we're doing makes David versus Goliath look like a fair fight, and we couldn't do it without you, the crowd. We are not venture-backed. We are a group of outsiders. My background's in biomedical engineering, proteomics, the study of proteins, protein engineering, really in the context of cancer biology, but more from an engineering perspective, actually developing new analytical methods and techniques for being able to interrogate what's going on in a cell, what's the cell signaling, called cell signaling transduction. But all this is very important because at the end of the day, the pathogenesis of diseases, they all end up converging on differential protein expression, through upregulating, downregulating genes, and stuff like that, right? So, and then I worked in investment banking, which was horrible. And that's how I met the founder, right? So neither of us, we don't come from this background. We don't come from this big pharma background. We don't come from, Silicon Valley background at all. And what we've been able to do without any venture financing is in complete defiance of the biotech paradigm. Biotech is a game of exclusivity. Small biotech companies are formed by ex-big pharma employees that have the purse strings, that have the connections. They're financed by venture capitalists in Silicon Valley. One of the reasons for that is the biotech CapEx is enormous. What I mean by that is the capital expenditure to get a drug development program up and running is huge. If you don't have about 25 million in the bank to start, you probably need to raise the money before you start doing any trials, right? Last thing you want to do is run a clinical study and not have the money in the bank. Biotech companies are beholden to venture capitalists and ultimately beholden to big pharma on the other end once the asset has been developed. What we've been able to do is in complete defiance of that. We're not funded by venture. We're 100% funded by retail investors. Through crowdfunding, raised about $22 million through all investments coming from accredited investors, some higher net worth people, total of about $34 million. Of that, I've raised about 25 of it. It's in essence, what we've been able to do is set a new standard and precedent for the way that biotech can function. And what I mean by that is we've demonstrated that drug development can be democratized and investing can be democratized. But let's focus on the drug development part. You see, if you're funded by an institution like venture, only certain drug development programs are going to get funded. Those are the ones that have ultimately are going to have an exit potential, an exit multiple, a final valuation that can support the fund's economics. What I mean by that, there are different fund economics, but basically every single investment needs to have the potential for a certain return multiple for it to meet the definition of the fund's investment thesis, okay? That means that only certain drug development programs that are developing drugs for diseases that have markets big enough to be able to justify the valuation are going to get funded. That means a lot of orphan indications, meaning drugs that diseases that only affect 200,000 or less people in the United States. Those are left to rot. Simply, the future economic potential of a drug that has 200,000 people as a total addressable market is just simply not viable from an economic standpoint for venture. So by creating this new model of biotech development and capitalization, we're allowing the people to decide what diseases get tackled and also participate in the economic upside by investing while we're still a private company, right? It's truly an amazing thing. And we really just stumbled upon it. And, Cytonics has arguably been the most successful biopharma company to do it. And, I get calls from all sorts of biotech CEOs asking, how are you doing this? There are so many small biotech companies out there that are single asset, just like Cytonics, and are looking for non-traditional ways to fund, even if they can secure venture capital. Like us, we don't want it. We'll never take a VC on our cap table. Why? Because venture capital is aggressive, and what I mean by that is they undercut your valuation, so they take a lot of your company. And oftentimes they'll install board members for oversight. If they don't like the way something's going, they'll have rights to be able to reorganize management. And ultimately, their economic interest is placed above everyone else. And what I mean by that is on the cap table, they usually own a class of stock called preferred stock. And preferred stock in this context often has a liquidation preference. So what a liquidation preference is a guarantee that the owner of that preferred stock gets a certain return before everyone else. Right? So they are guaranteed to make a return before any of the common shareholders are able to. We don't believe that's right. So the democratization of this investing in biotech is something that never existed before Regulation A and Regulation CF were really became a thing in 2012. So we're on the forefront of this. And to date, we've been able to fund a preclinical and clinical program in osteoarthritis, and we have decided to expand into oncology. And so you probably saw the update. Recently, we got a provisional patent to start exploring, our A2M variants, like Cyte108, in melanoma. And there is a strong body of scientific research to suggest that A2M is actually playing some sort of role in regulation of melanoma metastasis. Now, a causative mechanism has not been established. What has been established is correlative. We've been able to see differential A2M expression in melanoma where high levels actually correlate with better patient outcomes. Now, what is known about cancer biology and cancer pathogenesis is that proteases actually mediate metastasis. And that makes sense that A2M then could be a putative therapeutic in melanoma. So just filed that provisional patent, which allows me, priority has been set. We can now talk about it. I had a call with LabCorp this morning. LabCorp is a big contract research organization. The only other one on its same scale is Charles River Labs. And we've designed a set of 7 experiments in vitro and in vivo, meaning in petri dishes and in mouse models of melanoma, to establish a causative mechanism of our A2M variants and be able to start designing experiments that will inform human clinical trials. Now, osteoarthritis will always be the focus of this company. We've been an osteoarthritis company since day one. However, expansion of the development pipeline is de-risking the entire company from an investment standpoint. And it's also a really, really exciting thing to do. So this was a very strategic decision made by the board. We made it very quickly, but intelligently. And it comes at the perfect time. This raise is winding down. Some of the funds for this are going to go into those preclinical studies, which are not going to cost that much. We're going to be able to get some really meaningful data that will enable PCT patents, the real patents after provisional patents, and should secure some of those in 2026. So very exciting to start pursuing melanoma. We have some support from a local university. One of my colleagues is a melanoma oncologist, or rather a PhD that studies oncology. He's not a MD. And so we're going to leverage him to help design some of these experiments and help interface with this contract research organization to be able to develop some very informative and sensitive assays to establish this causative mechanism and create some parameters of future human clinical trials that will be able to support the actual approval of A2M and our A2M variants as a therapy for melanoma. So very exciting stuff. This really is on the cutting edge. And, there's something else I'm not going to say it yet. I'm going to wait. So the raise closes August 27th. We're probably going to raise money again. I can't really talk about terms or anything like that. We are going to raise money again. Fully funded through the phase one, going to get the melanoma up and running. The cost of the phase 1B/2A is going to be anywhere from 3 to 8 million. Those are the quotes that I've gotten from big contract research organizations, as well as a CRO that specializes in osteoarthritis. They're actually based in Denmark. So a relatively small amount of capital will be needed to be able to get that fully funded, well, rather have a cash buffer, right? We got about 2 million in the bank with another 2 million coming in. That'll be four. About 400,000 is going to be diverted to melanoma. So we're going to go out and raise a significant tranche of capital at a significant valuation that could very likely be the final dilutive event and get us through the phase 2B trial. So the capital formation strategy is very clear. Really in a tremendously healthy financial state. There are very, very few biotechs that can say, you know what, we don't need any more money. Right. But, the truth of the matter is you raise capital when you can, not when you have to. And so going out and raising the next tranche that could be the final dilutive event, I would really like it to be at a significantly higher valuation, is very likely the right move. So, we're preparing for this, but can't really, discuss terms until, this offering closes and, we make some filings with the SEC, which are going to happen before the end of this year. So all very, very exciting stuff. I'm not going to touch on, there's one other really cool thing we're working on. It's a little preview. It has to do with machine learning. We came across a treasure trove of data, of osteoarthritis data. It is the perfect application to take a foundational model and actually train it on this data and create a predictive analytic, right? Which will be the absolute state of the art. And it's something we would actually make freely available. So I've kind of given you, too much information about it. We haven't made a press release or anything about that, but it's underway. The project's underway and it's funded. So that's going to be really cool. I'll tell you what, like I said, we really shouldn't be able to do what we're doing, but we just keep going. And it's all due to the fact that we have all of these equity crowdfunding shareholders. I think at the end of this round, there's going to be about like 8,500. It's really incredible. And, we're really looking forward to being able to report, positive phase 1 data, be able to report, some positive interactions with the FDA in an upcoming Type C meeting, be able to get an investigational new drug application filed to be able to start that phase 1B/2A. Those are the upcoming milestones. In addition to the melanoma preclinical work and some of this predictive analytics stuff, which is going to hopefully optimize the phase 1B/2A parameters. If we can even improve the chances of a successful phase 2 by 10%, that's massive. And it'll set a precedent. And like I said, we want to make something like this publicly available, right? A lot of work's going to go into it. I've got some really talented AI engineers, but enough about that. So upcoming milestones, like I've said, very excited to get the database lock, which means that the statistician can go in and start conducting some of that analysis. And then the medical writer can write up the clinical study report. That database lock is going to happen here in the next couple of weeks. We're going to get the clinical study report out by, looks like early next quarter. And then that Type C meeting, that interface with the FDA in the early part of the first quarter 2026, and then start recruiting for the phase 2 trial shortly thereafter. So a lot of really cool milestones. Should expect to have some PCT melanoma patents in 2026 as well, and start fleshing out a full clinical development pathway predicated on this early stage preclinical work we're going to do with LabCorp. So I've talked a lot. Let's, I've answered some of these questions that have floated in, but there have been a lot. So I'm just going to go through and hammer these out. James asks, I've already invested, but I'd like to add my wife as owner or beneficiary. That's something I think you need to do on your end, like in a living will. I don't think that's something we really do on our end. Anthony asks, what's the latest estimated probability of return? How many X after we get an FDA approval? That can vary, that can vary very much. I'm going to turn to this slide. All right. So, how do you value a pre-revenue research biotech company where the assets are intangible, they're patents? How do you value the monetization potential of those patents? Well, you have to look at the future potential economic value of the drugs that are being developed over their viable economic lifetimes. Their viable economic lifetimes are very clearly delineated by the patents. So if you can forecast out the cash flows and then reduce those cash flows to account for the probabilities of failure in clinical trials, and also account for the probabilities of the or discount because there's an opportunity cost associated with any allocation of capital. You have to account for that. You apply a discount factor to the sum of the potential cash flows over that patent lifetime, and you come up with a value. And that value is an enterprise value. What the value of the company could be predicated on the future economic potential of its drugs in development, or in the case of Cytonics, its single drug in development, soon to be two once the melanoma comes about. So that's what produces this valuation curve, right? Now, the goal for the company isn't actually to get through approval. Our sweet spot is getting that phase 2 data and then either listing the company or partnering or getting acquired by another biotech. And so, this valuation right here could be something that's very reasonable. You have to understand that all these figures are derived from an Excel spreadsheet, right? And my projections, right? And I have to, disclaim that, right? These are all projections, right? Happy to provide anyone with that model, which I made very user-friendly. You can go in and you can toggle it and, put in different parameters, different assumptions, and see what gets produced. But this is what could potentially happen. And this is how a potential acquirer would go in and try and value the company. So JS, a great question. Is it worth, so basically his premise is that, we're setting a new precedent, right? Ventures out, the people are in. A quick flip isn't necessary because those demands for a return that venture demands is not there. It's still there, but not in quite the same way in terms of timeline and in terms of multiple. The demographic of the shareholders is fundamentally different. It's the people, it's patients, it's physicians, and there are a good number of people that really just want economic return, and that's fine. This is an investment opportunity. You should be looking out for money, right? But it's very possible that Cytonics remains completely independent. We keep funding ourselves via Reg A. We never subject ourselves to the public markets, which there's a lot of variability. I can talk about that here in a second. And we certainly never subject ourselves to big institutional private financiers like venture capital. And in doing so, we just become a full-fledged drug development company, and we have multiple drugs in development, a broad pipeline, and based upon approvals and partnerships and ultimately sales of approved drugs, we collect revenues and they could be distributed as dividends. That would be very, very non-traditional, but basically kind of a revenue sharing biotech. It's totally possible. Is it probable? Probably not, but it's certainly possible. And surveying the shareholders and seeing what they would like, it's certainly an opportunity. Look, it's a brave new world, and we're proving that every single raise. And so I'm totally open, to hearing what people have to say. Bella, when are you scheduling the next phase human clinical trials? How can someone volunteer? So tentative, obviously this is contingent upon, some FDA feedback in the next meeting. I would like to say we're ready to start recruiting in the second quarter of 2026. Email me, my email, let me just slide here. Is my email there? Excuse me. All right, well, I didn't put my email here. It's joey.bose@cytonics.com. Just email me your contact information. I just dropped it in the chat. And, we'll take care of you. We'll add you to the list. No guarantees. We don't even know where we're necessarily going to conduct it. It could be conducted solely in the United States. It could be conducted across the United States and Europe, or even back in Australia. Jared asks, I'm not going to hold you to, but what's the best guess probably success at each phase of development? So historically, the highest rate of attrition is at phase 2 at approximately 35%. Phase 1's around 70%. Phase 3, it goes up to, I think, about 60%. But what does that tell you? It tells you that the most difficult portion is efficacy, and proving that you're able to see efficacy at a meaningful level that is worth repeating at large scale in a phase 3 clinical trial. Because you have to remember, drugs aren't necessarily just abandoned because they failed to meet endpoints. It could just simply not be economically viable. And so, if a pharmaceutical company doesn't get a good readout, they'll make a strategic decision in phase 2 to say, hey, it's not worth, this is too difficult. It's better to shutter the program and reallocate capital to other things, right? So those are generally the historic. Let me see, I think the Q&A. All right, do we have anybody with any other questions? All right, well, this is recorded. Hope you enjoyed it. The deck is available on the website. I'll actually just type this in here, house.com. And, take a look at it. If you have any questions, you're always welcome to email me. And I just want to thank everyone for their support. Once again, this will be the largest raise. Looks like we'll converge, somewhere right around 7 million. Again, fully funded through the phase 1. Probably have about 4 million in the bank. So we're going to raise another tranche of capital, and I can't talk about the terms, but we're going to knock that one out of the park, and hopefully it'll be the last round of dilutive financing. So Jay asks, is it worth pursuing funding sufficient to complete phase 3 at this point? For the next round, no. And one of the reasons for that is we want to be able to back into evaluation, not certain say back into evaluation. We want to be able to substantiate evaluation that's not going to dilute the shareholders ridiculously. And raising capital right now for a phase 3, which could easily cost $100 million, would be simply too dilutive. We would need to do it at a different valuation, at a higher valuation. But the melanoma, adding some of that to the pipeline, that moves the needle. And where, like I said, there's a strategy, around backing into, or maybe I said that during the live chat. I didn't really talk about that. Oh, I didn't talk. I did the live chat right before this. So I didn't back into public listings and how all that stuff works. So it is relevant. It is relevant for you to figure out how are you going to make money. We're private stock. It's not liquid. Well, one of the traditional methods of providing liquidity to private shareholders is an IPO, an initial public offering. An IPO basically consists of paying in exchange to have a ticker and list the stock on that exchange so we can trade among market participants, some of which are professional market participants and other are you and me, right? When an IPO occurs and the stock is available to trade, all of the private shares would get dispersed to your brokerage of choice. For example, I trade equities, or I invest equities using Fidelity. There's Charles Schwab, T. Rowe Price. Actually, T. Rowe Price is mutual funds, but whatever, Vanguard. There's all sorts of brokers. If you don't have one, you get one set up. Our transfer agent, third party that administers the shareholder logbook, will disperse those funds. So basically, ahead of a public listing, all of our private shareholders get their funds dispersed to a broker who can then, sell the shares on the public market. What people don't understand is, NASDAQ IPOs, while they seem glamorous, right? A lot of founders, especially tech founders, they love this. You're going to go and you're going to ring the bell. It's a big deal. The truth of the matter is most NASDAQ IPOs are not Facebook raising $10 billion at a $100 billion valuation. You'll never believe this, but the average amount of capital raised in an IPO is $10 billion. You see, when a company IPOs, not only are they trying to give their shareholders liquidity, but it's a financing event. And so companies will sell treasury stock into the market as part of the IPO. Now, Most IPOs, when they raise a piddly $10 million, they're direct listings. What I mean by that is not underwritten. No investment bank has come in, diligenced the deal, assigned a share price, and taken an allocation, meaning assuming risk by buying some of the shares to be sold in the IPO, right? When Goldman Sachs and JP Morgan come in and syndicate an IPO, that's fundamentally different than a small little company getting a ticker and just listing their stock. You see, when an investment bank gets involved and they take that allocation, they assume risk, and they're going to resell your stock in the market. They're going to provide aftermarket support because they own the stock. So they're not going to allow the share price to tumble. When a company direct lists, here's what happens. They go in, all of the private investors, not insiders. Insiders have a lockup, usually 6 months. All of the private investors sell the stock because guess what? They want to make money. And fair, totally fair, right? But the share price starts to tank. There is no institution coming in to support the share price, to buy the stock in the public market. Then what happens is short sellers come in and they're just betting on the stock going all the way down. What is the result? The company raised their piddly $10 million, and now they're a penny stock. They will never be able to finance again without diluting their shareholders into oblivion, without basically capitulating and conducting what's considered a reverse stock split. Having the number of shares, doubling the share price, right? It's a complete capitulation. And once you've done that, you got a smear on you. You're not a serious company. You get delisted. There's all sorts of things. So it's not so glamorous to subject yourself to the capriciousness of the public markets, if you're doing, a little direct listing. So it is a viable exit, but it behooves companies to stay private as long as possible. But the consideration always has to be given to, well, how are the private shareholders going to cash out? Now, for venture, oftentimes they'll create a secondaries market where, and you'll see this for like SpaceX, there are brokers that buy large allocations of private shares from SpaceX employees and sell them in large blocks, but usually the minimum investment is like $250K. So it's only for accredited investors and relatively, wealthy people. So there is liquidity on what's considered a secondaries private market, but not for all companies, only for these big companies and only for these broker transactions. So all that's to say is that, a NASDAQ IPO, it sounds all well and good. Most of it is not nearly as sexy as you think, and it can absolutely destroy the cap table of a company. So what this means is that, as a CEO of a company, you have to, especially of a biotech company, where there are very clear catalysts and milestones upon which you structure your capital formation. The capital formation starts at seed and goes all the way to exit. And it might be an IPO, and that might be great, right? If you are a real company with a real development pipeline and Goldman Sachs underwrites it, you go in, you raise $300 million at a $4 billion valuation to conduct a series of phase 3 trials. That's legit, right? But you don't go to the public markets to raise $10 million and become a penny stock. You go to the Reg A market. And that's exactly where crowdfunding has filled this vacuum between venture and the NASDAQ. Most companies, once they run out of venture financing or can't get it at all and are, just funded, God, I don't even know how. If you don't get venture financing in biotech, you're basically screwed if you don't have, crowdfunding, right? Basically, your only shot is to NASDAQ. But with equity crowdfunding, we can stay private and raise $10 million, $20 million to do the research without subjecting ourselves to the public markets and to potentially destroying the ownership structure of the company. Our founder owns 18.9%. On a fully diluted basis, I project I'll own somewhere between 5 and 10%, which is phenomenal. This is not atypical. The company's been around since 2006. We have multiple dilutive financing, selling of stock, right? So what we're doing here with the equity crowdfunding, with the democratization of biotech, the democratization of investing, is allowing the company to proceed and create shareholder value without destroying the ownership structure until we're at a point potentially that's mature enough to be able to substantiate a IPO at a high valuation with aftermarket support, with clear use of funds that will continue to generate alpha and keep the share price going up, even though people are selling the stock. So you have to be intelligent about it. And truth of the matter is, you look at all these crowdfunding deals, and I don't want to speak ill of people, but holy shit. I mean, do people really know what they're doing? Half these founders, they don't know. Anyone that's technical, here's a big red flag. If your CEO is a PhD that's not run multiple companies, that has no Wall Street experience, they don't know what they're doing. Chances are. That is a big red flag. If you're looking at any early stage biotech, if they're an MD, they're a PhD, that's not a bad thing to have, but there better be MBA after that, or you should have a track record of entrepreneurship. If you don't, red flag, right? You see a lot of these companies, see biotech companies are listed on these platforms. I don't think they know what they're doing. I don't think they understand valuation. I don't think they know how to back into it. And what I mean by that is plan out your capital formation strategy, predict when your final dilutive event will be before the exit, potentially the IPO, and work backwards of what the valuation should be to be able to give your shareholders an honest return. What I mean by that is what valuation should the last money in come in at, so that when the stock either trades or gets acquired, that multiple is reasonable, and then work backwards from that. So some foresight has to be given into it. And the truth of the matter is a lot of these deals, I don't know where they're coming up with stuff from. But Reg A is an asset class is growing. In the first half of 2025, there was something about $680 million that have flowed, capital inflows into the market. That pales in compared to venture, which does anywhere from 100 billion to 200 billion a year. And obviously pales to the capital markets where the S&P's market cap is over 50 trillion. But it is a legitimate asset class unto itself, and the capital inflows are increasing. And I think some of that's due to the fact that there are better and better deals out there, like serious companies like Cytonics, where we actually know what we're doing, and have a strategy, and plan A, plan B, plan C. But I'm very confident that as the deal quality gets higher and higher and higher, which I've seen over the years, that it's just, rising tide raises all boats. And so we're very happy to be at the forefront of this. We're redefining the way the biotech can be conducted and financed outside of the grasp, of the tendrils, of the grip of big pharma and venture. We are probably the most successful biopharmaceutical company to do it. After we do this next raise in 2026, we will unequivocally be that company. We will set a new precedent, and extremely grateful for everyone that's supported us into this point, and have enabled us to do the impossible. So looks like we got a couple minutes here, but one more question. Okay, so if you guys need any tech support, just email me when it comes to placing the investments. I have to connect you with Start Engine. I realize that trying to transact, using IRAs and trust, these things can be complicated. Unfortunately, Start Engine, they have, these private equity associates that are supposed to be able to facilitate this. But honestly, I can't do anything on the back end. I'm an issuer, and all I can do is keep advocating for you. And I run it up the chain. And so as soon as you email me, I'll have it at the highest level. But yeah, please email me, joey.bose@cytonics.com, and we'll get everyone taken care of. And with that, I want to thank you guys again. Always a pleasure. Raise ends tomorrow at midnight, and looking forward to sharing a lot more updates. See you guys.